This video shows an example of facilitated diffusion or transport by the glucose transporter GLUT1. This comes from a paper in the Journal of Molecular Biology in 2017 by Yan. Here's a ribbon diagram showing the protein GLUT1. The membrane would be here. So this is a protein that crosses the membrane, and this current version is shown with it open at the top. Here's a second view of the same protein, again showing the membrane. And in this case, around the outside, they've shown an outline of the surface, the structure of the atoms, and then the ribbons of the alpha helices in the interior. This one's also in the open conformation, open to the outside, inside. And here they're showing glucose the size that we'd expect it to be. So glucose has come in through this open part. There are three parts to the protein: this N region, C region, and intracellular hinge. The protein works by keeping this N region relatively rigid. The C region can bend and hinge back and forth, back and forth. It hinges based on the connection down here. So this part moves back and forth, back and forth, back and forth. Here's the same image you saw before. Here would be the membrane. And if we zoom in, we'd have glucose approximately this size coming in and binding here. Once glucose is bound, this blue portion, the C portion, can shift in position. Notice that here we have an opening that's about the same size as glucose, but with this shift, that opening is much narrower. At the next stage is a further movement. Notice now completely closed here, narrow here, and here. So glucose is now held in the middle, and it's closed at both ends. I've added a little bit of darker blue line here because the light blue is a bit faint. The next stage, the transporter rocks a little bit more. This part moves out, and glucose is now free to leave. What makes this transporter specific to glucose? The R groups on amino acids, N, C, C in here. Here's the R groups. All of the reds here are oxygen, most as OH, hydroxyl. OH, 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 oxygen, OH, oxygen. In fact, these are double bond oxygen, 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 oxygen on these particular amino acids. All of the blues here are amino groups. The dashed lines are showing hydrogen bonding. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogen bonds holding this glucose in place. This transporter can sometimes move a few other closely related sugars, ones that have almost the same structure, but it's quite specific to a very limited number of structures. And that's because these are spaced just right to form hydrogen bonds well with glucose, but not others. In order to do the rocking motion, it either has to have a glucose bound there, in which case this paler blue part can rock back and forth, can close or open, or that middle needs to be empty, in which case it can do the rocking. So if there's more glucose on the outside, glucose will enter when it's in the open conformation from above, bind in the middle, shift, and leave on the inside. Equally, if there's more glucose inside the cell, glucose will move out. The direction of glucose movement is dependent upon the abundance of glucose. So this transporter will move glucose in any cell in both directions. The net movement will depend on where there's more glucose. How might we sketch this to get the concept? Here it is open to glucose on the outside. Here's the intermediate stage when it's closed on both sides. And the other stage when we've opened up to allow glucose into the cell. What makes it specific? If there's glucose in here, it can shift. And if there's glucose in the middle, it can shift in this direction. If it's empty, it can shift back and forth. So it can move glucose across and shift back, empty. Move the glucose across and then shift back, empty. What it can't do is make that same change if there's something else in there. Something else in there and the hydrogen bonding is wrong around the edge, and that prevents this from tipping back and forth. So we're going from stage to stage, but we know we can go in either direction, and you also want to remember we also can go in these directions when it's completely empty. So we can do this. We can also do this.